thank you for taking the time to come to our immunoprecipitation workshop today. So my name is Callie Anderson. I'm a technical specialist here at Proteintech, but today this workshop will be led by Dr. Christian Linke-Winnebeck, who is a principal scientist at the Chromotek division of Proteintech Group. So Christian studied biochemistry in Germany and France and did a PhD in structural biology in New Zealand. And during his years as a postdoc in New Zealand and Germany, he grew so fond of nanobodies and antibodies as tools for research. They joined Chromotek in 2016, and Chromotek is now part of Proteintech Group, and it has been since 2020. And Christian has participated in the development of several different nanotraps for immunoprecipitation. So he really is an expert on this topic and really well placed to lead this workshop today on immunoprecipitation. So before I hand over to Christian, I'll just do a really brief introduction to Proteintech as a company, just in case we have any new faces that are not aware of us as a company yet. So Proteintech is a leading manufacturer of antibodies, immunoassays, nanobody-based reagents, and cytokines and growth factors. So we really have our products under three main product lines. So under our Proteintech brand, we have our antibodies and immunoassays. So this includes antibodies to over 13,000 targets, our new antibody labeling kits, and also ELISA kits and immunohistochemistry kits. Under humankind product line, we have our cytokines and growth factors. So these are all human HEC293 cell expressed. And because they're expressed in a human cell line, this means that these proteins have native folding, post-translation modifications, and glycosylation, which means these show really superior activity in cellular assays. So we have research use only and GMP grade products on this product line. And we also have our Chromotek product line. So these are nanobody-based reagents, and this will be more of a focus for the talk today, so I'll definitely let Christian give you a full introduction to the nanobody-based reagents as part of this talk too. So at Proteintech, we really celebrate our success by that of the researchers who are using our products. And it was really fantastic to meet a recent milestone of 200,000 product citations worldwide. So we're really happy about that. And we're also really happy about a recent award that we received just last year. And this was a Researcher's Choice Award. So this was voted for by researchers such as many of yourselves who are really happy with the service and the quality of our products. So that was just a brief introduction to Proteintech, and I will be moderating the chat box and the Q&A and trying to answer any of your questions coming in and gathering some together for the end of the workshop for Christian to answer to. So with that, I'll hand it over to you, Christian, to get on with the workshop.
Fantastic. Thank you, Carly, and good afternoon, everybody. Thanks for joining us today for this workshop here. It's a pleasure to be able to talk about IP. I gather that some of you maybe routine users of IP, while others might be only looking into the method for their own research questions. So I hope to have something for everybody, like from the complete novices to the expert users. So if in parts it might be boring to you because you're already an expert user, please bear with me. I can offer something to you as well. Otherwise, any questions, please put them in the chat for Carly. So just get us on the same page, talking about immunoprecipitation. I'm not going to say immunoprecipitation for the rest of the talk. I'm just going to say IP. So as you all know, IP is a method in biochemistry to isolate and enrich a protein of interest. So you usually start off with a protein extract, like the lysate, for example, lysate of cells or tissues, and then we really isolate one protein and maybe its binding partners. For that, generally, they use antibodies, that's the immune part here, which bind then to the protein of interest, and antibodies at some stage immobilized on what we call a matrix but generic expression. I mean anything that's a bead, agarose beads or magnetic beads, which allow us to isolate this one protein from the others, wash away all the others we don't want, and then in the end analyze the one protein of interest to us. Quite quickly, probably most of you know that already, the workflow of an IP is first, you have a lysate of some sort. Then we need the binding of an antibody nanobody to a protein of interest. We wash off anything that's not bound to my nanobody or an antibody. And then we have to get the protein off the antibody in the end to analyze it. And then we can use something like SES-PAGE or Western blot or mass spec to look at our protein. So one of the important things is, of course, the antibody we use for our IP. It's the one recognizing our protein, and ideally only our protein, not anything else. There's a few things to watch out for. One thing, first of all, is please go for validated IP antibodies. Not every antibody is suitable for IPs. And I'll tell you about this in a moment. Also, it helps if the antibody has a high affinity, and it should definitely be purified, because it's a range of different purification grades for antibodies. Something that is purified via the antigen is great. Protein A and or G purification often works. But if you have, for example, a serum that contains all the other antibodies against all the other targets, then it's not going to work, because all the other antibodies are going to poison your beta matrix and you will see no binding. The other aspects, like if you want to use polyclonal antibodies, here both work, the pros and cons. If you think about how you recognize your protein, you can either use antibodies directly against your target or put a tag onto your protein of interest, like GP tag or flag tag or MUC tag or something like that. And if you think about the type of beads you're going to use for your IP, there's a whole range of different bead types. Some of them are based on agarose or cephros, which give them a very porous nature. Others will be solid little beads. These mostly magnetic beads, for example, dynamo beads. These have no pores at all. One difference here is the one bead based on agarose, you have to sediment them using centrifugation, while you can use a magnet for magnet beads, of course. Also makes a difference depending on the type of your protein you have, because if you have a very big protein or a huge protein complex, it might not be able to diffuse into the pores of a porous bead. But however, all the antibodies will be in the pores of these beads. So, but if you exclude it, then there will be no binding. But first, more about antibodies. Why is it important to look out for the right antibody? Because there are antibodies that are great for Western blot. There are antibodies that are great for IP. Some antibodies are great for both, but sometimes antibodies only work in Western blots, for example. And the reason for that is the part of the protein they bind to. In Western blot, for example, you have denatured proteins because you boiled all your proteins in laminate buffer, they're all denatured. The structure is gone, and the antibody can bind wherever it wants to within the protein. The situation is different for IP, where you're interested in native proteins in the native fold, because often you're interested in binding partners. For that, your protein has to be active and properly folded. So the antibody can't bind anywhere. You only can bind to the accessible surface of your protein. So the difference here is often that antibodies purely for Western blot are generated by immunization of peptides, while for IP, we often use whole intact proteins for immunization. So especially what's been done at Proteintech, but also at the other companies. And the second difference is for Western blot, antibodies can actually be quite bad when they still bind, because you have a membrane here where you have a small patch with your domain of proteins, and the antibody, with two binding arms, can bind twice. So even a weak antibody can bind twice, and the binding adds up. It's what we call avidity. So you have actually quite strong binding. While in IP, your antibody, in this case, attaches bead, and yes, you have two binding arms, but each binding arm can only bind one protein, and these two proteins are separate. They're not on the membrane. Though the protein can go off easily. If affinity is bad, you lose your protein again during washes. So you need high affinity for an IP antibody, just because of different types of binding. So yeah, many companies will tell you what they validated the antibodies for. Some companies don't. At least for Proteintech, I know if you look at our website for antibodies, it will tell you if you've been if you've tested them for IP or not. Then go for the IP tested antibodies. If you have a good antibody, next question is how to immobilize on these beads. Here again, got different options. In some cases, you can buy pre-conjugated antibodies that are covalently bound to beads, like here on the right side here. This is of course an advantage because an antibody will stick to the bead, it won't come off again. And I will show some more examples later. But many other times you have a naked antibody, and then you can capture the antibody to a bead using protein A and or G. Protein A and G proteins derived from gram-positive bacteria, and these proteins are naturally binders of antibodies. It's a great thing, because then you could just buy protein A or G beads, and their protein A or G is covalently coupled to your beads, and then can capture your antibody, because as shown here. However, you have to watch out for the type of antibody you're using. Because you might have, if you have been working with protein A or G before, you probably have seen some of these tables here, which give you the different specificities for protein A and G for different antibodies. If you look here, rapid IgG, right, then you see in both cases good binding, but the story might be different for like some mouse IgG1, for example. And as you know, if you have a mouse antibody, mouse monoclonal, it's a high chance it's a mouse IgG1, because that's the most common subtype for mouse monoclonals. If you use, for example, chicken IgY, which is not uncommon, then you have no binding at all by protein A or G. And to show you the different type of binding you can see here, a couple of years ago, we made some experiments in my team here, where we used protein A and G to capture antibodies on a BLI system. BLI is biolayer interferometry. It's a bit similar as like Biocore or SBR, so you can measure binding affinities. So in a nutshell, you've got a sensor, it's put protein A and G on sensors, and then you can have your antibodies in your soluble phase, and you can look at the binding of these antibodies to protein A and G in this case here. So for example, here, got a rapid IgG, rapid antibody. You can see first a binding phase, nice strong binding, and then the so-called dissociation phase, which is just looking at the stability of the complex of the antibody and protein A and G. And what you see here is we get very strong binding. Here, protein A in blue, protein G in orange. The antibody doesn't come off, it stays on protein A and G. So if we have a similar situation as for protein A and G beads. However, if you use mouse IgG1, it's a very common subtype for mouse antibodies. You see here for orange protein G, again, we got strong binding, but then within a few minutes, the antibody comes off protein G again. This is even worse for protein A, that we see hardly any binding at all. You can see a little blip here. We can come on, says hello, and then it's gone again. So here, with the fraction of a second, you have protein A is off. Sorry, the mouse IgG1 antibody is off your protein A. And for comparison, your mouse IgG2A, again, a very second common mouse monoclonal subtype. You get some good binding for protein A and G, but again, over the time course, you lose this binding. Protein A and G will get off the beat. So it's actually an example here. If you have used protein A and G and use mouse IgG2A, and they give you a good long wash, you will lose the antibody. So you have to be kind of watch out which resin you're using and which antibody with protein A and G. Just as a side here, sometimes you don't have to think about protein A or G because they actually beat with both protein A and G. Then you can just put whatever antibody on it, but still, even then, you'll have the effects shown here. Certain proteins, certain antibodies will come off your beads over the time. An alternative here is something I want to show today. The alternative is our nanobodies. As you all know, our normal antibodies, which are shown here, which we find in mice or men, or similar animals, where we have this antibody complex of 2 heavy chains, 2 light chains, making up this 150 kilodalton antibody, 2 binding arms. Here, the Fab fragments shown here, left and right. And you get the same antibodies in camelids, but camelids, which are animals like camels, bacterian camels, dromedaries, or alpacas or llamas, actually quite related to each other. And these camelids have additional heavy chain antibodies. So they have the normal antibodies, but all these heavy chain antibodies shown here. And these heavy chain antibodies don't have a light chain. In addition, like a part of the heavy chain here. So the antigen binding domain is really small. It's just this tiny domain here, which corresponds to this domain over here. Technically, for immunologists, it's a VHH domain here. And we often call them nanobodies because it's a bit of a trademark name, like sellotape or something. Important thing is here, we can isolate these antigen binding domains here, these VHH or nanobodies, can express them by themselves, and then have tiny antibodies, these nanobodies, which have the whole functionalities of antibodies, strong, high affinity binding of antigens, or a protein of interest, but it's 1/10 of the size of normal antibody. The nanobody is about 15 kilodaltons big, which is 1/10 of the size of 150 kilodalton normal IgG. And there we have some advantages. I'll show them in a minute. One of the advantages here is these tiny antibodies, nanobodies, are quite stable, which makes them very well suited for IP. And we can covalently bind these nanobodies to beads, and then what we call a Chromotek or nanotraps, nanobody traps for the purpose of IP. The most popular one is a GFP trap, shown here. So in the following slides, I'll often refer to our GFP trap and to IP of GFP, but all the advice on IP will also hold true for other more conventional antibodies or other traps. To give an example of our nanotrap, in this case for GFP, here we should compare the pull down or the IP of just GFP by our nanotrap and a conventional antibody. The first thing that is quite striking is if you do an IP with our nano trap, our GFP trap, you only get the band in the SES page here. You want to see there's an SES page, so full staining of the bound fraction. There's only one protein, GFP. If you do the same thing with conventional antibodies, you will see additional bands, of course, because if you have a conventional antibody and pull it down using protein A or G beads, it's not much you can do about it. You will see the antibody chains in your SDS page here, the heavy chain and the light chain. It can be quite annoying, especially if your protein of interest is similar size as the heavy chain and light chain. And it's always a problem for Western blots because sometimes you have to, for example, if you use a secondary, the secondary antibody might recognize or will probably recognize your IP antibody as well, which gives nice strong bands and a signal for your heavy light chain, but then the protein you're looking for might be masked by these heavy light chain. So here, so that's a big advantage here of our nano traps. The nanobody is covalently bound to the beads, so it stays on the beads when you boil up your samples, and you only get your protein of interest. Although, especially in the case of the GP trap, it's really high affinity binding. If you're into biophysics, here the KD is about 1 picomolar. So if you do a pull down, it's a GFP here in the Western blot in the input, but you won't see anything in the unbound fraction. You get a nice strong bound fraction, while with a competitor GFP antibody. Yes, you will see pull down, but you will see that lots of protein remains in the unbound fraction. That's because affinity is not high enough. So nanobody is great tools. It's also a great tool if you do mass spec analysis because it makes a huge difference for your background peptides. If you have a small 15 kilodalton background protein or 150 kilodaltons. And you have nanobodies, nanotraps for a range of tags. For example, that's what I can nanotraps really recommend for. Like if you're using tags like GFP tag, MTR tag, MIG tag, B5, or our spot tag. And then I really recommend doing these IPs using our nanotraps, followed by Western blot using a different conventional antibody, which is a great experiment on the setup. If you pull your antigen with one nanobody and then have an orthogonal antibody recognizing your protein in your Western blot. So whenever you see something now, it's your protein, of course, because you have two independent antibodies working on your protein. If you're looking into endogenous targets that you don't want to tag, which of course are legitimate, then you can recognize a collection of antibodies, of conventional antibodies, but still here I would recommend to use one antibody for IP and then use a different antibody for the detection within your Western blot to again have this orthogonal detection of your protein of interest. So now we've got our antibody beads or protein A, G beads, or we use nanobodies, your nanotraps here. How do we go about our IP? Just to summarize again, for an IP, you need first sterilize it of some sorts. We need to bind our protein of interest using our antibodies. And we need to immobilize our antibody on beads. Then we can wash off any unbound stuff. And then finally, you need to get the protein of our antibody again to be able to analyze it. Here, I wish I could give you like a fully fledged protocol that works for any IP you ever want to do. However, unfortunately, I have to say that the optimal IP protocol will really depend on the antibodies, but also on your question. So it really depend also depend on your type of tissue or cells or whatever you have. So, and then often you have to optimize each step for the perfect IP. This is, I know it's a bit sad to hear, but at every step, things can go wrong, but then it can be optimized as well. So I'm going to talk about what you can do in the following few minutes. First step is, of course, making a cell lysis, because for an IP, you need soluble proteins. So, as a side, we're going to talk much more about it. If you're interested in membrane proteins, for example, then you would have to solubilize your membrane protein first before you can do anything about it. Assuming a soluble protein, the method of lysing your sample really depends on your sample again. So here, if you have cells, you're lucky, because cells, especially human cells, can be lysed quite easily. If you're using plants, it gets more difficult, because you have to you can go back to good old liquid nitrogen and pestle mortars. But here, depending on the sample, trust your own judgment, your own experience with your samples, and find the right ways to lyse the cells and also tissues as much as possible. Important thing here is a bit different from other methods. We need a buffer that will allow the proteins to stay in the native state. So we can't use any buffers that are common for example, for RNA purifications. But these buffers are usually quite destructive and will unfold any proteins you have in your samples. So please go something physiological. Go with something physiological. Having said that, even a so-called physiological buffer like PBS, it emulates the salt situations in blood, but not necessarily within cytosol of cells. So you might have to adapt the buffer and salt concentration and detergents to your sample, either a tissue sample or to your protein of interest. Regarding the amounts, if you're working with human cells, for example, it's quite easy. You can use 10 to the power of 6 or 10 to the power of 7 cells. It's a good starting point. If you're working with plants, you need about 10 grams. If you have a tissue, 500 microgram of total protein are all good starting points. But again, this might depend on your actual sample. What's come to all those is it's best to use a fresh cell lysis. Because and also avoid freezing of the lysis. It's perfectly fine to freeze a sample before lysis. It's of course quite common. I mean, you collect, for example, mouse samples over a couple of times and like store these samples in your freezer. It's fine, but then once you start lysing these samples, you should go straight ahead to your IP if you can. I know sometimes it's not possible, sometimes, but then I would recommend to reprocess your sample, for example, to make sure there's no debris in your sample. Because what often happens is if you analyze it, if you leave it in the freezer, freeze it, thaw it, a good way to kill proteins, and they will aggregate, and they will form little aggregates. And these are really bad for IP because they will poison your IP with lots of background. So in that case, if you have to use lyse it from the freezer, make sure you either centrifuge it very hard to sediment any debris, or filter it using, like, for example, using a 0.2 micrometer filter, which you always should do for the fresh lysate as well, but it's worth repeating for old lysates. But if you can, use fresh lysate, because things happen in lysates. You know, lysates have protein that aggregate, protein that age. Then we have kinases and phospholases and so on. They all will change your proteins in there. So please use fresh lysate. So once you have lysate here, nicely fresh and without any debris, you can add your antibody. What I would recommend here is add your antibody just for 1 hour for binding. Some people like to go overnight because it's something they're used to from Western blots, for example. But don't do that for IPs, because again, you've got a lysate here, and this lysate is not dead yet. You know, you've got all the proteases, phosphorolases, kinases, and they all continue to work within your lysate, so they change everything. So make sure you keep the timing for your antibody binding as short as possible. That's one thing here. If you use one of the nanotraps, you can just add the nanotrap to the lysate and incubate that for an hour, and you can go to the Bosch steps. If you use conventional antibodies, there are 2 ways to do things. You can either first add your naked antibody to the lysate, incubate it for an hour, and then add protein A or G beads. Or you can kind of pre-assemble the complex out of protein A, G beads with the antibody first, and then put this complex of beads plus antibody into your lysate. Both ways have advantages and disadvantages. Again, it's one thing nicely was that's nice with our nanotraps. Just put them in. I don't have to think about that. So yeah, once you put your antibody in there, make sure your lysate is kept mixing. It's very important. I would recommend here is some kind of end-over-end rotation. Don't go too fast. Don't vortex your sample, because again, vortex is a perfect way to destroying proteins. But something slow and steady end over end would be great. Like the good old wheels, you can find some labs. They still work perfectly. Then make sure your samples in this range here, 200 to 500 microliters, the good range for an IP. Sometimes you have to go higher, for example, for plant material, but if you can, try to stay with this volume here. Also very important, stay as cool as possible. Don't freeze your sample, of course, because there's no mixing, but go for 4 degrees, because again, lysates change, they do things that aggregate, that with all these enzymes in there, and 4 degrees will inhibit or slow down all these processes. If you stay at 4 degrees in 1 hour, hopefully you have a good snapshot of your lysate. And yet, as mentioned before, go for a mild buffer because you don't want to denature your proteins. You want to have them in your natural state and well folded. So let's assume we have our antibody with the target protein on our beads. You can start washing the beads to get rid of all the proteins, debris, lipids, whatever you don't want in your analysis. Here you can go a bit harsher for the binding buffer. You can, for example, add detergents or go higher with the salts, and it really depends on your protein. But try to stay quick here. Just go for a couple of minutes of washing. I go, for example, for now traps we often do is have like our Eppendorf tube, the beads on here in the middle, at the bottom. You just add some buffer, like 500 microliter of washing buffer, add it on the beads, a quick rinse, then put it back to the centrifuge to settle the beads and take off the supernatant again, or to the magnet. This usually is enough. And then just do this 3 to 5 times. And so the total wash step should take only like 15 minutes or something like that. And they should be done. And then you should in total have used like 1 to 1.5 mil of washing buffer. Of course, if you extend washing, go longer, or more steps, or have bigger volumes of your washing buffer, you would just lose your antibody if you're using protein A or G beads, or it's a good chance of losing your protein of interest from your antibody unless you use something really high affinity binding like the GP trap. And if you're interested in core IP, where you're interested in the binding partners of your protein, also a good chance you're losing your binding partners, because often many protein interactions are actually of relatively low affinity. So yeah, stay quick, be quick, and don't overdo it for washing. One important thing here is if you do, for example, if you do 3 washes, one important thing is to transfer your beads to a fresh tube for the last wash, because there are proteins binding to your tubes, not coming off before you. And then if you boil them in limiting buffer, they might come off again and they'll look dirty. So in the end, let's say we have a nicely washed protein on your antibody beads. Even to get this protein off the beads again. You get quite a few options for elution here. You just want to do a Western blot. Of course, you go for denaturing elution. You can just add SES page sample buffer, or it's often called lamin buffer, to your beads, and boil your beads, and the protein will come off. And you can use supernatant for SES page. If you don't want to use lamin buffer, alternative often is acidic elution. For example, using glycine at pH 2.5. pH 2.5 or pH 2 works for most antibodies. There are some antibodies that are not acidic, not sensitive to acids, then you can use NOH. Importantly here, just make sure you neutralize your buffer afterwards, because pH 2 or pH 11 can proteolyze your protein, so you disrupt a few things. So make sure if you after elution, you add some other buffer to get the pH back to normal. If you're interested in your native protein, so you don't want to denature it, because even if you use pH 2, you will denature it. Then one option could be the use of a specific protease. So for that, this works well if you have a tag. For example, if you have a GFP tag, you could put a protease recognition site between the GFP tag and your protein of interest. For example, you could use a thrombin site, or here I've mentioned the TAV protease, that's a tobacco edge virus protease. People that work with protein purification will know this protease. This works also for elution from IP beads. In some cases, you can also use native elution using a competitive peptide. For example, if you use a flag tag, so a short peptide as a tag, and you can add free flag tag peptide, or the same for spot tag, for example, to elute your protein competitively. And then you will have your protein still folded, so it's still active. You can use it for other types of analysis, like for example, for biochemical or enzymological analysis. Just a quick word here, because sometimes people think it would be nice to have an antibody with really high affinity, but at the same time you can elute it using peptides. However, the problem here is that elution, innate competitive peptide elution and high affinity are then kind of mutually exclusive. The reason for that is here that if you look elution using peptides, you have a situation like this. You have your protein on the antibody, on your treats, then you add an excess of peptide. And the thing is, the peptide doesn't displace the antibody, the protein. What happens first is the protein comes off the antibody, and then the peptide can bind. Because you have an excess of that peptide, it's a higher chance of the peptide binding to the antibody than the protein. But for that to happen, you need a certain dissociation rate. So the protein can't bind too strongly to your antibody to be able to get off again, which means that you have a lowish affinity. Whenever you can elute a protein using peptides, the affinity won't be as strong. However, then at least you can elute it and can have nicely folded, nicely active protein. Unfortunately, you can have it both. If you're looking into mass spectrometry for your protein analysis, another option would be on the on-beat digest. So this is, I know there are many options of many ways of doing mass spec. This is geared here towards LC-MS fingerprinting analysis. For example, if you want to do a pull down or see what of certain protein and looking into binding partners. The most efficient method, in my opinion, here is to do the pull down and then use trypsin on the beads to digest your protein on the beads, so you combine elution with the trypsinization. For that, you just do your IP as usual, but make sure that for the last washing steps, you exclude any detergent, because detergent and mass spec doesn't quite work together. And then you can elute your protein using trypsin in a mixture of urea and DDT and so on. And then you have got peptides here in supernatant, which you can purify for your mass spec analysis. If you're interested in that, we've got a protocol on our Proteintech website for the GFP trap, for example. So let's assume you've done all these steps, and we want to do a analysis using SES patient Western blot. I just want to recommend not only looking at your whatever's bound to your beads, but also at other samples such as your lysate and the unbound fraction. For example, here for the STS patient Western blot, we highly recommend to put on a sample of your input, that is a small sample of your lysate, and then also a small sample of the flow through, the unbound stuff, and then your bound fraction. So ideally, in the SS page, you have input and flow through, because you have lysates, you have lots of bands, and then one band, or depending on your protein, more maybe for your bound fraction. But in your Western blot, you should also see the protein was there in the input, and ideally it's not anymore in the unbound fraction, so you're completely depleted on the beads now, and then we get a nice strong band, which hopefully is bigger than your input band for your bound fraction. This all seems very obvious, but using these 3 lanes here is really helpful if you have any issues, or if you have to improve or optimize your IP. It's a very good way for controlling your experiment. I will show you some examples here the following few minutes. But before that, we also recommend controls, as usual. You know, we all know the controls are very important, but it's definitely the case for IPs. And one thing would be a positive control, which is easy if you have a tag like GFP, because then you can include an IP that just have your GFP tag, just because GFPs are protein by itself, of course. So you can do an IP of GFP and can double check your beads and your Western blot detection are working. If you don't work with a tag, you might be lucky to have your protein of interest in the form of a purified recombinant protein, which you can also pull down to double check here that the beads and or antibody and your Western blot is working. Then, even equally as important is a negative control, which you can really recommend, especially if you started working with a new antibody or new type of beads. Because this negative control here will make sure you can recognize any unspecific binding. In that case, if you use a tagged protein, for example, it's GFP tag, you can do a pull down from a lysate where there's no tag, just your protein of interest, to make sure the protein of interest is not sticking to the beads themselves or any other protein. If you're looking for an endogenous protein, it's a really good idea to do a pull down from cells, knock down your protein of interest. For example, you can do a siRNA or CRISPR-Cas knockdown, and ideally, if you have knockdown or knockout, you should see less or no of your protein of interest in your pull down. And only then you know the band you see in the other pull down is really your protein. The second thing you could try, especially important for protein A and G beads, is to pull down with just your beads, or if you use nanotraps, to use another nanobody. It doesn't bind to anything which is within your lysate, to make sure you can detect any proteins that just bind to the beads, which could be important, for example, for protein A and G, because protein A and G are antibody binders, and there might be antibodies in your sample, depending on what, for example, if you have a lysis of a mouse tissue, there might be antibodies in this tissue here, and then can detect that using your protein A and G beads. So you know, okay, if you just use protein A and G, you will still see bands. So these bands are something different from what you trying to look at. If you have these problems here, if you have unspecific bindings to beads, you can do what we call pre-clearing. So you do a depletion step first. So you have your lysate, add your protein G beads with no antibody to make sure anything that's binding to these beads is depleted first. And then in the second step, you start your proper IP with the actual antibody. And for nanotraps, you can do a similar thing with your with a different nanotrap that doesn't bind. If you're looking for GFP fusion proteins, you could do a M-cherry nanotrap depletion. Also, which also can happen is that sometimes you have antibody binding proteins in your lysate tissue or lysate. Again, for that you could use an isotype control, like an antibody of the same class and species, to deplete these antibody binding proteins. So, a couple more minutes from my side. I would like to talk about some troubleshooting because, as I mentioned before, unfortunately, there's no ideal protocol for all IAP experiments in this world. You have to adapt your protocol, and you'll often see issues. And we're going to talk about these very briefly here. We go over to questions. The first thing is you have a pull down and you see too many bands here, like in this case here. So, and unless you have a very highly complex with many subunits, it's probably unlikely to want to see that. So reason for that might be that you have use too much lysate. It can be a problem because you have a certain amount of debris, or your protein gets degraded, or you have protein aggregation, or the lysate was frozen and thaw again. And as I said, freeze thaw is a good way of killing proteins. Get rid of that. Make sure you optimize your IP setting, your IP protocol for your cell type, for your lysate, to make sure you get the right amount of cell material. You might have to optimize your buffers to make sure your protein of interest is happy, because proteins have certain preferences for pH and salt concentrations for detergents. You might have to adjust that. Make sure you're working at 4 degrees for the binding step and maximum binding of 1 hour. I have to do pre-clearing, as mentioned before. And then, of course, there are other things like you might have to add protease inhibitors, but it's something you'd recommend anyways. The other problem might be the washing step, that you just have some lightly bound unspecific binding. This can often be get rid of by increasing salt concentration. For example, if you use NaCl, increase that to 500 millimolar, or you can increase the detergent, and make sure to use a new fresh tube for the last wash step, as mentioned before. The other extreme is if you don't see anything at all in your bound fraction. But then here it's really important to have these controls, as shown before. We also look at the samples of your input and your flow through, because in the Western blot here we'll see, okay, your protein was there and it's still there in your flow through, but not in your bound fraction. So it wasn't bound. So there could be several reasons. One reason could be that your protein is insoluble or aggregated. It could be a mixing, could be a problem if a wheel stood still for a while, or you didn't have enough beads, or as mentioned before, if your protein is so big, it doesn't get into the pores of your porous beads, then you might have to look at another bead type. So here you can try to solve this problem by working at the construct. For example, if you do an overexpression or transfection of a tag protein, you might have to think about your tag positions, or because these might have an influence on the protein expression or the solubility. And then you can look at the cell lysis again. Buffer is really important here, or even how you lyse the cells. Make sure binding is thorough by end-over-end rotation. And yeah, might have to test another bead matrix like magnetic beads. And if you're really unlucky, you don't see anything at all, again in your bound fraction, but you don't see anything in your Western blot either, not even for the input fraction, then the reason might be that expression levels of your protein of interest are so low, you can't detect them anymore. Or your protein is insoluble, so you lose it. It's a step when you get rid of your debris, like when you do the filtering of a lysate or centrifugation. Then you have to make sure if it's in debris, you have to kind of find out why it's in debris, either because of membrane protein or it's just heavily aggregated. So aggregated is kind of not even soluble at all anymore. Then again, you should work on your conditions for lysis or the expression construct, or the redesigning construct, or look into detergents or something like that. Or if you're really unlucky, I have to look into another antibody for Western blot detection. Maybe just doesn't. It's just the wrong antibody, not suited for your type of Western blotting. Well, that could happen here is that you see a protein in your bound fraction, but then if you look at your Western blot, you see, okay, actually the amount of protein in your bound fraction is less than in your input fraction. So somehow your protein stays bound to the beads and doesn't get eluted. For that, I can recommend to make sure you use enough of your elution buffer. For example, if you use SDS sample buffer, Lamley buffer, make sure there's use enough, because the beads, especially if they're porous, have their own certain volume of buffer in there. But if you add elution buffer, you would dilute your elution buffer just because the buffer in the beads. So you make sure you've got enough in there, and you have to make sure you mix the beads nicely to give your protein a chance to elute. So I would have one more slides here, but running out of time. So something, I guess, Kali with the other slides here, but let's get to the end. Remember, choose the right nanobody or antibody for your IP. Not every antibody is suitable for IPs. It's very important to know. How you prepare a sample really affects your results. And unfortunately, each step can be optimized. It should be optimized for the best results if you want to have some really reliable results. And don't forget to include your controls, because they will enable you to interpret the results and to find any problems. So Give back to Carly. She's, I think, been collecting your questions. And hope you can answer the questions now.
Hi, sorry, my camera's back on now. Yeah, thank you. There's been lots and lots of questions in the chat. I've answered a few.
Oh.
I think we've had many questions, and what we will do is we can collate them all and maybe do a bit of a frequently asked questions responses and send that out with the email as well for some of these. But I think that we can answer a few. So I picked a few sort of common ones which were coming up, Christian, which maybe you could help with. So a common question that we had was, what about if you have to look at the endogenous protein and there's no antibodies available which are validated for IP? So are there things to look for in antibodies that you might want to test in this context?
So it's a bit unlucky. So if you have a protein of interest and you don't have a validated antibody, the one chance is that some of the antibodies that are not validated against IP might still work. So if there are any antibodies at all, it's probably worth sort of testing them. But then, of course, you have to go through all the controls, make sure the antibodies really just binding your protein of interest. It's doable. The other option would be the other option would be to go for tags if you can, depending on the system you're using. With cells, it's quite easy to go to make an expression construct, to transfect your cells with an expression construct of your protein of interest with a tag on, like GFP tag, flag tag, MIG tag, spot tag. There is different pros and cons for these tags. And then you use a generic anti-tag antibody for pull down. And you can also, in the modern world, you can also use CRISPR-Cas to check. If you don't like the transfection, you can use CRISPR-Cas to change or add a tag to your protein of interest within the genomic level, which is also doable.
Yeah, that's a good idea. I think having an idea, it's that kind of pre-planning, isn't it? If you know there's not going to be good antibodies available for your very specific protein, try to work that in the planning stages of a project. It's always good, the tags. Okay, so I had another question which I thought was a good one. What would you constitute as a harsh versus a mild buffer? Obviously, when we're talking about all these different buffers, there's lots of questions about what buffer would you recommend for X? And I think, in my experience from doing technical support for this, it really varies depending on the protein of interest. It's difficult, isn't it?
Definitely. It's really like one of the things we can say depends. I'm sorry to say that. However,
what would you say is harsh versus mild?
Generally speaking, for example, for me, harsh would be something that contains urea, for example, or gadolinium hydrochloride, because these compounds are really meant to denature proteins, and they're, as I said, often used, for example, for RNA preps, for example. This would be harsh. Certain detergents like SDS can be used for certain systems in IP, but for most antibodies, they'd be quite destructive and just really denature your antibody as well, and then it's dead. There are exceptions, for example, the GFP trap. People use it for IP and then wash it up for really harsh washing using SDS and urea. It can work, but it's quite an exception. For 99% of antibodies, doesn't work. So then the other things, like for harsh washing or harsh conditions, extreme salt concentrations, like no salt at all, it can be quite harsh. If you go like to 1 molar of salt, like NaCl or KCl, again, they'll be quite harsh as well. And anything in between really depends on your sample, on your protein, on your tissue, on your cell. They has to be tested, I'm afraid.
Definitely. It's a lot of trial and error, I think, with these different buffers.
Unfortunately, yes, yeah.
So I had a few questions that came up about really, I guess, elution conditions. So one person had asked, what elution conditions would you recommend if the protein of interest is degraded easily?
Mm-hmm.
And what should you avoid, perhaps?
Yeah. Again, depends a bit, but assume your protein of interest degrades at room temperature. We can do like have your do your IP quickly, then add your Lamy buffer, and then boil it, and then put it on a gel directly. Because the question is here, why does your protein degrade? If it's some kind of protease contaminations in your sample, which often happens, and then if your protein is quite sensitive to your proteases, it would be worth first adding protease inhibitors, of course, and then doing the dilution step is using Lamy buffer, because they should destroy and denature your proteases as well. Having said that, some proteins are sensitive to Lamy buffer, actually. So I don't know, I've done it before, but then you might have to apply the temperature you're boiling your protein in, because like usually boil your protein at 95 degrees. For some proteins, you have to lower the temperature to 70 degrees or even lower. Just make sure the SDS can work. It's magic to nature protein and bind it.
Sure.
Then if you don't want to dilute your protein in Lamy buffer, if it's sensitive, again, add protease inhibitors and make sure you keep your protein at 4 degrees as long as possible, as much as possible. Keep it on ice if you can. Just make sure, because the lower the temperature, the lower any proteolytic rates. Find your sample here. Of course, don't freeze it, but
there was a few questions about the protease inhibitor, which is suitable to use. So I know certainly using a protease inhibitor cocktail, which contains lots of different inhibitors. Do you have any other recommendations?
Yeah. Yeah, cocktails are great, I think, because they give you a range of inhibitors. Otherwise, ETA, for example, is a good inhibitor because it will inhibit metallic proteases. Then you have the classic serine protease inhibitors like PMSF, which can make your own mix if you want to. But if you can afford it, I would go for the cocktails, especially in your lysate.
Definitely. We had a few questions about boiling the eluted samples, boiling for elution if you have membrane proteins. So I know that we can, as you said there, reduce that temperature when you're doing the elution, but maybe increase the elution time perhaps to try and prevent any sort of aggregation that membrane proteins can be prone to in some cases.
Yeah. For membrane protein, unfortunately, yes, you have to adapt the boiling protocol. It's one of the many cases, yeah, where boiling at 95 degrees will give you a pretty bad sample.
Yes.
Yeah. So yeah, definitely reduce it, and then depending on the antibody you have, yeah, you might have to extend your time of elution. But for many antibodies, 5 minutes in SDS at 50 degrees should still be enough.
Sure. I had a question which I think is it's a good one. It's an important one. So about the recommendation of using 2 different antibodies for IP and detection during the Western blot. So could you outline the sort of key benefits of using 2 different antibodies for that?
Yeah. The thing is, if you use the same antibody for pull down the detection, what might happen is that you get some unspecific binding for the pull down, but then if your antibody is unspecific or has some then you would pick up the same bands as your detection antibody. You would think, oh yes, it's my bands here. That's what I'm looking for. However, if you use an orthogonal antibody, which is a different antibody for your Western blot, this antibody should rebind and should have another pattern of unspecific binding. So ideally, the one antibody binds the main your protein, then some other stuff, and then the other antibody should only bind your protein because it won't recognize the other bands. So you should have a clear band only for your protein, and then you can be quite certain the band you see now is your right protein.
Yeah, of course.
Yeah, that's the main advantage. And we have this before that, for example, if you use fusion proteins with GFP, then the customer is saying, oh, that's not my protein here because it's the wrong size. However, if you pull it down with a GFP nanobody and then use a different antibody, and you still got a nice thick band. And it is probably GFP and your fusion protein, but just not in the format you like it to be. For example, it's degraded, for example.
I see.
Easier picked up if you use 2 different antibodies. So you can be sure about it.
Yeah, absolutely. And I think as well, if you use antibodies from the same species for the pull down and the Western blot detection, this is where you really stumble into the problem with detecting those denatured heavy and light chains on your Western blot from the
sorry, a very good point,
antibody.
Kali. Yeah, it is. Yeah. Thank you.
No, of course, and that's where the nanotraps has such great benefits. You don't have any issue with that as well.
Good point. Yeah.
Okay, I think we've reached the hour mark here, so I want to thank everyone for joining. I hope that you found it useful, and I know there's still a lot of questions. So what we will do over, let's say the next week or so, we'll take a good look at these questions and put together some answers, and we can either reply individually to some people, or I think we might put together a bit of a FAQ answer so that everyone has access to that information. We also have a lot of really excellent blog articles which people might find interesting to do some more further reading of their own about all of this, so we can highlight those in the follow-up email too. So with that, I'd really like to thank Christian for an excellent workshop, and we will be in touch soon with the follow-up. Just to highlight, I said in the chat, we will definitely send the slides and also a recording of the webinar today too.
Fantastic. Thank you as well from my side. Thanks for listening in.
Great. Thanks, everyone. See you.